Hi, my name is Denise Blanchette, and I work for the Maine Department of Environmental Protection in the Invasive Aquatic Species Unit. I welcome you to a whirlwind tour of using eDNA to detect zebra mussels and Asian clam in Northeast lakes. Zebra mussel and Asian clam are present throughout the U.S., and there are several programs in the western U.S. that use eDNA to detect established populations to develop management strategies. In 2017, the Maine boat inspector data, there were 1,117 out-of-state boats inspected. They came from 40 different states, 4 different provinces. 603 of those boats came from infested waters. And since people are the primary vector of spread for invasive mollusks, and their villages can be invisible to the naked eye, it seemed prudent to find a way to detect these species early. In 2018, I began a conversation with Alison at the University of New Hampshire about using eDNA for the detection of invasive mollusks. Our goal was to build off the experience of the West and develop a method for early detection. We began with some simple questions. Is there a method? Are the species in the database? Has someone developed a primer? Will they give us advice? With a yes to these questions, we moved forward. We put together a team of partners interested in the project: New Hampshire Department of Environmental Services, New York and Vermont Department of Environmental Conservation, and the Lake Champlain Basin Program. Once the lab validation was done, the partners developed an approach to sample collection, lab analysis, and study design. We selected 2 sites with both species, Lake George in New York and Lake Bomoseen in Vermont. We selected several sites with only 1 species. Great Pond in New Hampshire has just Asian clam. Lake Memphremagog and Dunmore in Vermont have zebra mussels. And we used Tawny Pond in Maine as a control. Samples were collected using a peristaltic pump with 47 millimeter filter and 1.5 filter. The decontamination procedures were done with 50% bleach solution between all sites, and all sampling included a field blank. We collected 5 1-liter filtered samples at 3 locations and increased the distance from the primary infestation site to get an understanding of what the distribution of the eDNA signal was. Samples were collected in May, July, and August. Samples in the field were stored on ice and then shipped with blue ice within 24 hours to the University of New Hampshire for analysis. So for brevity, I will show you the results of the zebra mussel and Asian clam in Lake Bomoseen, and then discussed a few unexpected results. Lake Bomoseen has a very robust population of both invasive species, zebra mussel and Asian clam. And the initial sampling results for Bomoseen were what we pretty much accepted. We had positive results for zebra mussel and Asian clam at all sites for all 5 samples. The signal was strongest in the early season and got less as we went through the summer. And it was strongest at the source than it was further away. One of the unexpected results was in Lake George in New York. So Lake George in New York has a low density population. And when they do discover zebra mussels, they immediately remove them manually by divers. So in consulting with our partners at the USGS, we discovered that the primer that they use is less sensitive. Since they're not looking for small populations, it was not an issue for them. So in September, we switched our primer to the more sensitive one. However, we knew from our data that September was not an optimal time to detect zebra mussels and Asian clam. And as a result, even with the new primer, we got a very low detect result at one site on Lake George. In 2019, most of our sampling was canceled due to COVID-19. However, we were able to get one sampling event on Lake George and at a known location of zebra mussel and Asian clam, and we were able to get a positive result there, taking larger volumes of water. The other interesting result from our 2018 sampling was that on Lake Bombazine. So Lake Bombazine, we got an early detect in spring, well before the temperatures were at 12 degrees C. And most of the literature told us that 12 degrees was the prime time for detection due to spawning. We were detecting it at an earlier temperature time than what the literature was. So what did we learn? We learned that Northeast is not the same as the West, and that we got some different results than what some of the other things are. So using a primer that's relevant to the type of work you were doing was very, very important. We also learned that sampling early in the spring was the best, that the signal is strongest closer to the source, that when infestations are small and non-viable or recently removed, they're hard to detect, but you can detect them if you take larger volumes of water. It's going to be a job of determining and analyzing cost analysis for eDNA versus traditional survey to determine what we do with that. So future work, we want to resample the low concentration sites on Lake George, Memphis-Maybog, and Lake Dunmore in the early spring, taking larger volumes of water and focusing on areas with known colonies to develop a low concentration detection methods. We also want to sample Lake Bombazine weekly to create a time series data set in which we can see peak detection times and maybe get a better idea of if there is a spawning difference between what's going on here in the Northeast and other places. We're also hoping to compare with USGS, we're hoping to compare methods between the traditional manual method of plankton toes and looking at veliges versus the eDNA method. And then we're hoping to use these data to create an occupancy model in order to put together early detection programs for these invasive mollusks. And then not the least, but also very important, we hope to develop a manager's decision SOP for the Northeast so that managers, when they have a positive detect, know what that positive detect means and how to respond to it in a positive way. I really want to thank our partners in this project, Alison Want at the University of New Hampshire, Meg Moodley at Lake Champlain Basin Program, Steve Pearson from New York Department of Environmental Conservation, Kimberly Jensen from the Vermont Department of Environmental Conservation, Alan Sepulveda and Eric Erickson at USGS, and Amy Smagula at New Hampshire Department of Environmental Services, and a special thanks to Ian Vove, who was our citizen volunteer and helped out with some sampling. Thank you very much.